encodes the protein itself, but you have also regulational information which controls when, how, and where it should be expressed. And this regulatory information is not very clear on the basis of the DNA sequence. So what people do is they just take a piece of DNA which encodes the gene, the protein, and they just put it into a mouse and study where it is expressed. Has it sufficient information surrounding this structural information to get it expressed properly, then you have the complete gene. If not, then you have to extend the DNA on both sides to get it expressed in a normal way. If you do that, then you can use these strand genes to study the biological consequences. And you can combine that with gene knockouts and with other mutations. First of all, and primarily, these mice are used to make models for human diseases, and that's what we do for cancer. And so you can, then mutations that you find in a certain disease, in our case tumor, you can use those mice to confirm that mutation is causally related to the onset of the tumor, or causally related to, for instance, the onset of diabetes or any other disease. Subsequently, you can use these models to try to set up a therapeutic intervention in these models. That saves a lot of work doing these preventions, these therapeutic intervention studies in humans. So you can select your therapeutic intervention protocols first on the basis of what you do in mice, and then subsequently you can do that in humans. And that's what we also do in the Netherlands Cancer Institute. First we create the models for certain diseases, and subsequently we test them for therapeutic intervention. Now there are several methods to make genetically modified mice. Basically there are two methods. The first method is just straight zygote injection. That's the most simple one, and it was already done some 30 years ago. And you have a cloned piece of DNA which encodes your DNA, your gene of interest, and you just directly inject it in the pronucleus of a zygote. A zygote is a merge between the oocyte and sperm, and when the sperm enters the